estuaries are under threat and are being transformed by urban, agricultural and industrial pollution, and in many cases even removed completely by hydrological alterations. However, these ecosystems are very important for humankind. They protect us, feed us, and connect us. That's why it's so important to understand how different pressures affect them, and to try to limit the ecological damage done as far as possible. The sediments at the heart of the estuary is the home of millions of microorganisms that, although invisible, are crucial for the ecosystem, forming the base of its food webs.
The high functional diversity of these microbial communities make them good indicators of the health of the ecosystem.
Through the MICROMON project at ASTI, we use their DNA as a tool to try to understand and monitor the health of the estuary ecosystems.
We also study how these microorganisms interact with each other to better understand what they do, their functional roles, and how they react to things like urban and industrial contaminants. Some species are more sensitive than others to these kind of impacts.
Thanks to modern sequencing techniques, a small sediment sample like this is more than enough material for extracting environmental DNA that allow us to study the biodiversity of the entire microbial community present.
This DNA can be used to amplify a specific region in the genomes of the species present, called a taxonomic marker. This marker is later sequenced to study the diversity of the sediment.
We focus on the microbial communities because they're essential for the biochemical cycles, with interesting and partly unknown functions. Together they form the basis of the coastal food webs.
In the MICROMON project, besides estuaries from the Basque Coast, we analyze sequences from other parts of the planet, from South America to Asia and from Europe to Australia, with different levels of disturbance.
We look for organisms that can serve as indicators of estuary health, not just locally, but globally.
We have seen that microbial communities from estuaries that share similar properties like climate, salinity or health status are often very similar to each other, even if they come from different continents.
For instance, Queensland in Australia and Florida in the US have similar microbial communities, just like Tasmania and the Bay of Biscay.
This shows that environmental DNA sequencing can be a powerful tool for evaluating the health of the ecosystems such as estuaries.
There are many challenges, like standardizing the methodology across different labs and countries. But in the end, it's clear that we can use these techniques for environmental monitoring in a way that is fast, accurate, and relatively simple.